Hello students, in today's class we are going to learn about mathematics of growth and different phases during bacterial growth. Okay. So first we'll discuss about different phases of growth. So as you can see diagrammatically, when we plot a graph of the log number of viable cells versus time during a growth phase, during a growth experiment, we will be able to see that it is divided into four main phases: the lag phase, the exponential or the log phase, the stationary phase, and the death phase. Okay, so the lag phase is the first phase during of a growth phase, and this is the initial stage where the where there is no growth happening, and we just supplement our medium with small group of cells, and the cells are basically trying to adjust to the nutrients that are present in the medium, and they are synthesizing all the different components that are required, all the different enzymes that are required so as to use up the nutrients, and it is basically preparing, it is also expressing all the proteins that are required for replication and cell division to happen during the next phase. So it is, you can say that it is a preparatory phase wherein the cell is not actually dividing, but it is preparing for cell division and growth in the next stage. Now, the second stage is the exponential phase or the log phase, where logarithmic or exponential growth takes place. So, here at regular time intervals, the cells are dividing by binary fission and they are doubling in number at the end of each generation time. So here the metabolic activity is high as DNA, RNA, cell wall components and other substances necessary for growth are already generated and the cell is dividing. Then the third phase is the stationary phase. So during stationary phase, the overall population of the experiment will remain constant. So this is because the rate of cell division is equivalent to that of the rate of cell death. Now, the cell death will begin because of, first of all, depletion of nutrients and accumulation of certain toxic waste products, because of which the death will begin. And during this stage, the bacterial growth will basically reach a plateau or a stationary phase, and there is no overall increase in the cell number or the population. So also spore formation will begin by those bacteria which have the ability to form spore during the stationary phase. Okay, and then there is the last phase which is known as the death phase or the decline phase. During this stage, the nutrients become very limited, and there is a high amount of waste products that are accumulated. As a result, the cell death rate increases exponentially. As a result, the population number decreases rapidly. So only spores will be able to sustain or survive during the death phase, which will, whenever the favorable conditions will return, they will be capable of multiplying again. So this is about the different phases of bacterial growth. The next growth curve that you can see over here is known as a dioxic growth curve, and you will see that in dioxic growth curve there are basically two lag phases. The lag phase 1 followed by the logarithmic growth phase. Again, there is a lag phase followed by the second log phase. Then there is a stationary and a death phase. So there are these two lag phases that are appearing in a dioxic growth curve. So we will see this type of growth curve when we are making use of two different substrates, and especially when one of the substrate is glucose and other substrate can be any other sugar. So glucose is basically the preferred sugar for metabolism, and whenever glucose is present, the organism will only express enzymes for utilization of glucose. So that is why the first lag phase, where the organism is adjusting, expressing all the enzymes used for required for utilization of glucose. Now, when all the so during the log phase, you can see there is exponential growth because the enzymes are synthesized and the bacteria are just multiplying. But when the glucose has exhausted, the microorganisms will take a pause and they will understand that what is the other nutrient that is now available. Then, through the regulatory mechanism, it can understand or sense presence of the other sugar, for example, lactose. Then, during the second lag phase, it will make all the enzymes that are required for utilization of lactose, and then it will enter into the second log phase or the second exponential growth phase. So that is why we will be able to see these two short lag phases in growth curve, which is known as dioxic growth curve. Okay, so next we are going to learn about mathematics of growth and what are the different equations that we need to learn. So this is more relevant with the exponential phase of the microorganisms, wherein the microorganisms are dividing at a constant interval of time. So they are basically doubling in number at a specific length of time, which is known as the generation time or the doubling time. So if we assume that the original cell population is 1, then as represented in the table below, suppose if 20 minutes is the generation time or the cell division time, then at 0 time, there are no cell divisions that have happened. So the number of divisions are going to be 0. And the population will be as the original population, which can be represented by the expression 2 to the power 0, since the cell division number is 0. So n represents the number of divisions. So this value will come out to 1. At the end of one cell division, which is generally 20 minutes in case of bacteria like E. coli, the cell will divide once. So the cell population at that time can be calculated by the formula 2 to the power 1, which comes out to 2. Okay. So in this way, you can see that at every division, which is happening at regular time interval, the cell population is doubling in number. So initially there was 1, 2, 4, 8, and so on. So this type of growth is known as exponential growth. So these observations can be expressed in the form of an equation for generation time, in which n0 will represent the initial population number, nt will be the population at time t, and small n is the number of generations in time t. So the results in the table can be represented as nt, that is the population at any time t is equivalent to n0, that is the initial population multiplied by 2 to the power n, where n is the number of generations. So if we take log of the equation, what we will have is log nt is equal to log n0 plus n log 2. Rearranging this equation, we will have the value of n, that is the number of generations, is equal to log nt minus log n0 divided by log 2. We know the value of log 2 as 0.301. So substituting for it, we reach the equation n is equal to log nt minus log of n0 divided by 0.3. Now the rate of growth during exponential phase in a batch culture can be expressed in terms of mean growth rate constant, that is represented by K. This is the number of generations per unit time, and it is often expressed as generations per unit time. So by formula, we can write as K is equal to number of generations per unit time. So it is N by T. So substituting for the value of n from the previous equation, we have K is equal to log nt minus n0 divided by 0.301 multiplied by T. Now, so basically the time it takes for a population to double in size is known as the mean generation time or the mean doubling time, and is represented by the symbol G. So when the time required for the cell division is one generation time, at that time, the population, the given population is going to be double that of the original population. So we can say that when time is equal to one generation time, the population is going to be twice that of the original population. So substituting for NT in the above equation, we will get k is equal to log of 2 times n0 minus log of n0 divided by 0.301, and we are going to substitute for g instead of p, because when the time is one generation time, the population is going to be twice that of the original population. So simplifying the equation, we have log 2 plus log n0 minus log n0 divided by 0.301. So these will get cancelled out and these will get cancelled out. So what will be left with k is equal to 1 divided by g. So we can say that k is inversely proportional to that of the generation time, or mean growth rate constant is inversely proportional to that of the generation time. So there is one example of a problem. We'll try to solve this. The question says that if a bacterial population increases from 10 to the power 6 cells to 10 to the power 9 cells in 10 hours, then find the value of n, k, and g. Okay. So to first of all, to find the value of n, we need to use the previous formula. N is the number of generations, and the formula is log nt minus log n0 divided by 0.301. So from the log nt is the population at time at the end of the experiment, that is after 10 hours. So here it is 10 to the power 9 minus log of 10 to the power 3, that is the initial population, divided by 0.301. So after simplification, this will be 9 log 10 minus 3 log 10. Value of log 10 is 1. So simplifying, we'll have 9 minus 3 divided by 0.3, which comes out to 6 divided by 0.3, that is 20 generations is the value of n. Now the second task is to find the value of k, and we know that k is equal to n by t. Okay, so from the previous equation, we know that. So substituting for n over here, we'll have k is equal to 20 by t is 10 hours. So your answer is going to be 2 generations per hour. Okay. Now coming to the value for g. So g is inverse of that of k. So g is equal to 1 by k or 1 by 2, which is going to be 0.5 hours per generation. So in this way, you can calculate the value of n, k, and g. So that's it about mathematics of growth. I hope you like the content. Kindly like, share, and subscribe to get direct updates for the uploaded videos. Thank you.